Okay, great. Well, it's a pleasure to be here, and thanks Naranjan and Mihai for inviting me. I guess I'm the token talk in this session to try to explain how we've been trying to, although we're doing a lot of metagenomics and sequence analysis, sort of complement that with some experimental techniques for dissecting the microbiome. And I'm gonna focus in particular on one particular area that we've been interested in for the last few years, which is the role that the microbiome plays in changing the outcome of the drugs that many of us are taking. Okay, so I want to start by pointing out that if you're like me, you usually think about chemists as sort of nefarious people like Walt White and Jesse Pinkman making these toxic drugs in unsavory conditions. But it turns out that humans are actually pretty bad chemists, and we should really think about chemists like this. So the microbial world has really perfected chemistry, and they're capable of performing many different biotransformations that can't be performed by us, either in the lab or by our own enzymes. And in particular, the microbiome contributes a variety of different reactions that are important for health and disease. And those include helping us to digest our diet. They break down dietary plant polysaccharides and proteins. They produce many useful small molecules like essential vitamins and amino acids. They metabolize many of the endogenous compounds that are found in our bodies, so they contribute to the circulation of hormones like estrogen. They modify many of the bile acids that are found in the gut. And the reactions that we're particularly interested in is how the microbiome detoxifies, or in some cases actually activates, xenobiotics, or foreign compounds to the body. And that includes drugs, antibiotics, as well as what's called dietary bioactive compounds, so small molecules that are normally found in our diet that have sort of drug-like effects. And so research over the last few decades has identified dozens of different interactions between the microbiome and xenobiotics, and I'm just going to summarize a few ways these interactions can play out. So you can think about a drug or a prodrug that you're ingesting, and as it makes its way through the gastrointestinal tract, there's a variety of different things that can happen. And in some cases, compounds are directly metabolized by the gut microbiota, and that can involve turning on a prodrug, inactivating compounds, converting drugs to toxic byproducts that can contribute to the side effects of a medication, or in many cases, the drugs that we take have antimicrobial effects, and this can be intentional in the case of trying to rid ourselves of unwanted pathogens, but it can also be an unintended consequence that can affect many of the resident members of our microbial community. In addition to that, there's evidence in the literature that suggests that colonization by gut microbes actually impacts the way our own body deals with xenobiotics. And so that has been shown in two different ways. One is that colonization changes the expression of our own genes, both in the intestine as well as the liver. And then also these microbes are producing small molecules that make it into circulation and can actually siphon away our own enzymes, reducing or changing their ability to metabolize drugs. Okay. And I want to just point out that although biochemically this phenomena has been described for quite a while, it's featured in some prominent textbooks, but we still know essentially nothing about the underlying microbiology. And this is just a review that we put together of these 44 reactions. I don't expect you to read these, but these are just 44 different drugs that we know are metabolized. And I just want to make the point that of these 44 compounds, today we can only link 3 of them to a bacterial enzyme. So in the other 41 cases, we have no idea how these are being metabolized. And we can only link 5 of these 44 to any specific type of bacteria. So we know that the community as a whole has this activity, but we don't know who is responsible. And then obviously this could just be the tip of the iceberg. No one has systematically screened our collection of compounds, and so we have no idea how many of the remaining 7,000 or so drugs and drug-like molecules are influenced. Okay. And so I want to just briefly mention a couple techniques that we've been using in the lab to try to understand these interactions, and then I'll go into a little bit more depth, and hopefully this will give you a sense for the various experimental approaches we're using. So one of the things that we've been doing is sort of the, I guess, cheapest or fastest way to answer some of these questions. And basically, it's incredibly simple. So we'll collect a sample from a healthy human donor. Typically, this is a fecal sample. And then we just bring it under conditions that allow for the survival of the bacteria that proliferate in the gut. And that's typically just anaerobic conditions, 37 degrees. And then we expose these sort of cultures to a variety of different conditions. And then we assay the results in a variety of different ways. And so I won't go into the details, but we've been developing some methods for flow cytometry that allow us to measure bacterial cell damage and activity. We also use sort of standard 16S sequencing that allows us to look at changes of the community over time. And then we've been using metatranscriptomics or RNA-seq to look at expression changes in response to these compounds. And so last year we published a study where we looked at a panel of 14 different drugs. And this included 6 drugs that we know that are meant to target our own cells, but are known to be metabolized by the gut microbiota. And then we also screened a panel of antibiotics. And I'm just going to briefly point out that we were able to show that for each of these compounds we could find genes that were differentially expressed in the presence of the compound relative to the control. And that allows us to sort of compare the transcriptional response to this panel of drugs and look for things that are in common within a class of compounds and so forth. But I'm just going to, for the sake of time, sort of zoom in and talk about one particular drug that we've been very interested in. And it's a member of the cardiac glycoside shown here, digoxin, and then also its very closely related compound, digitoxin, that both showed a similar transcriptional response in this assay. And so we've been very interested in digoxin as sort of an initial case study of a drug that's metabolized by the gut microbiota. It's one of the world's oldest drugs. It's been used for hundreds of years for heart disease. It's a natural product that comes from this beautiful but deadly plant, the foxglove. And it's called a cardiac glycoside due to the fact that it has these 3 sugars. And so today, even though this drug is incredibly old and toxic, it's still widely used for heart failure and cardiac arrhythmia or irregular heartbeat. Its mechanism of action is due to the fact that it inhibits sodium-potassium ATPases that are expressed in the heart. And it's really challenging, I'm told, for clinicians because the therapeutic range is incredibly narrow. So doctors are told that they have to hit 0.5 nanograms per mil of the drug in the serum to get its effect. But if they go over 2 nanograms per mil, the drug starts to induce severe toxicity. And so this digoxin toxicity has been well characterized for many years, and it involves damage to the gastrointestinal tract. It can induce the same problems with the heart that it's used to cure. And then it has some more idiosyncratic side effects like blurry yellow vision and halos around lights. So as a microbiologist, we're very interested in digoxin because it was shown back in the 1980s that it's inactivated by gut microbes. And so this reaction is relatively straightforward. This unsaturated lactone ring here in digoxin is just reduced to the downstream product called dihydrodigoxin. And this single reaction inactivates the compound. So dihydrodigoxin cannot bind to its target in the heart. And so clinical studies identified that a subset of patients produce this compound at high abundance, and that this metabolite is unique to microbes. It's not produced by our own tissue. In addition to that, it was shown that if you administer a variety of different broad-spectrum antibiotics, you can elevate the amount of digoxin that makes it into the blood. So that's consistent with this idea that antibiotics are suppressing the reduction of the compound. And then John Lindenbaum at Columbia went even further to show that you could actually screen a panel of bacteria from the gut, and they identified just a single bacterial species that performs this reaction named Agarthala lenta, shown here. And this was really attractive to us because it's sort of a natural example of a single pair between a compound and one species of interest. Okay. So the other interesting thing about Elenta is that unlike many of the bugs in the gut, it's not consuming sugar. Instead, it thrives off of the amino acid arginine. And so if you grow Elenta in the lab and just add more and more arginine, in the solid line you can see the level of growth of Elenta increases. But what happens at the same time is that the amount of the drug that's metabolized decreases. And so in the dashed line, we've just quantified the amount of digoxin that's reduced. And you can see at low levels of arginine, this reaction occurs, but at high levels, it seems to be suppressed. And so we wanted to just exploit this difference using RNA-seq to try to look for genes that were upregulated in the presence of digoxin, but downregulated by arginine. And so we took a variety of different cultures of Elenta, and fortunately we have a really nice fully sequenced type strain genome. And so we just looked for that, and I'm just going to show you one operon where we saw a clear signal as we expected. So again, we're just scanning along the genome of Elenta, and the peaks here are proportional to the number of sequencing reads. And you can see in the absence of the drug, this operon is off. Whereas when we add digoxin, it comes on hundreds of fold higher. This was really exciting for us because Elenta encodes about 14 different bacterial cytochromes, and 2 of which fell into this operon. And so these are enzymes that could potentially be using digoxin as an electron acceptor. And so I'll refer to these now as the cardiac glycoside reductase operon for the rest of the talk. The other exciting thing about this was, just as we hoped to see, arginine downregulated the operon. And so again, this is by QRT-PCR. In the absence of digoxin, there's no expression. When we add digoxin in low arginine, it comes on, in this assay, almost 20,000-fold. And then that's downregulated about 2-fold in the high arginine condition. And we saw that occur in both exponential and stationary phase growth of the bug. The other thing, so the challenge, I guess, in working with Elenta is that we don't have any bacterial genetics, so we can't sort of definitively prove that these genes are responsible. And there's a variety of issues that make it challenging to clone these genes into heterologous hosts. And so we chose sort of the natural history approach, which was just to compare many different strains of Elenta for their ability to reduce the drug, and then try to link that to their whole genome sequence. And so we started out by looking at just 3 strains of Elenta. We've now increased this, although I'm not going to show you the data, to 15. And what we found is that the type strain was capable of reducing the drug, but these 2 other strains were not capable of performing this reaction. And if you compare their whole genomes, you, just as you would expect, see that these other 2 strains do not carry these 2 genes. So we next wanted to ask if we just moved to a complex situation where we had a whole mixture of organisms present, if we could use the abundance of this gene to predict drug metabolism. And so we performed, again, this type of ex vivo assay where we collected samples from 20 different people, incubated them in the lab with digoxin, and just measured how much was reduced. And we found that you could sort of stratify people into 2 different categories. So some people leave most of the drug intact, and we call them low reducers. And then there's high reducing communities that, for most of them, they metabolized all of the drug we exposed them to. And then if you just quantify the abundance of the CGR operon in these samples, we saw that, just as you expect, on average, the operon was found at a higher abundance in the community in the high reducing individuals. And you can sort of calculate the precision, sensitivity, and specificity of this assay, which, even in this early form is doing pretty good. And so this suggests that potentially we could use these individual enzymes as sort of a biomarker for microbial drug metabolism. Okay, so the next question we had was whether or not we could use this information that we have about Elenta's metabolism to think about ways that you might actually prevent this reaction in humans or other animals. And so the idea was that we knew that in vitro that we could use arginine to suppress metabolism. And so we wondered whether or not we could just modify the amount of protein in the diet and change the amount of drug that was metabolized. And so for this, we turned to gnotobiotic isolators, where we're able to raise animals under sterile conditions If you haven't seen these before, they're just vinyl bubbles where all of the air is filtered, all of the food and bedding is autoclaved, and so it's a sterile environment that you can maintain mice from birth until death without exposure to any microbes. And then you can introduce individual organisms and study them in these controlled in vivo systems. And so what we did in the initial experiment was to just take 2 different groups of mice. They were all colonized with the type strain of Elenta. And then we split them onto 2 controlled diets, one that had no protein and one that was 20% protein. And what we found was that on the high protein diet, we saw increased levels of digoxin in circulation. And the same was true in terms of the digoxin that was excreted in the urine. And so this is consistent with the idea that the increased level of protein in the diet is actually blocking Elenta's metabolism of digoxin, increasing the amount that makes it into circulation. But there's a problem, I guess, with this experiment, which is that it could be that diet just has an unknown effect on how the host deals with the drug. And so we wanted to control for this, and so we repeated this experiment using, again, the type strain, but then one of these control strains of Elenta that lacks the CGR operon and leaves the drug intact. And again, we found that in mice colonized with the type strain, we saw a significant difference, but we did not see that in mice colonized with the control strain, and the same was true in urine. Okay, so our sort of working model for how this is occurring goes as follows. We think that certain individuals might be primarily colonized with non-reducing strains of Elenta that leave digoxin intact, and so in that case, the level that you're given will reflect what you get in circulation. Whereas other individuals may be primarily colonized with these reducing strains, and they are able to convert the drug, but again, that's dependent on the concentration of arginine in the lumen of the gut, which could be influenced by dietary protein, but possibly other sources like microbes or the host itself. And then I didn't show you the data for this, but we have some early hints that other members of the gut microbiota may talk to Elenta in a way that drives its metabolism of digoxin. And so this means that if you, depending on the composition of the gut microbiota, Elenta may this reaction may occur much faster or stronger. And that may increase the amount that's metabolized, but again, that could be suppressed based on the availability of arginine. Okay, so just as a quick conclusion, hopefully I've convinced you that we've made a little bit of progress in understanding digoxin, but obviously that's just one drug of the thousands that are currently listed in Drug Bank. And so I think there's many questions that remain unanswered in terms of how the microbiome interacts with xenobiotics. We don't have a good sense for how many other compounds of these 7,000 are metabolized. We have really very little understanding of the underlying sort of core mechanisms for xenobiotic metabolism in the microbiome. That's been well established in the liver and the intestine, but we would love to identify sort of the cytochrome P450s or the core xenobiotic metabolism gene families for microbes. We don't know how dependent these things are on other factors. I mean, I think we have initial evidence that diet matters, but presumably host microbial interactions may dictate the outcome of these interactions, as well as many other microbial and environmental factors. And then finally, we hope that down the line, this could be used as sort of a proof of principle that by focusing on these individual reactions in the microbiome, we may be able to make recommendations relevant for personalized medicine. And so I just want to acknowledge all the great people who've worked on this, worked in the lab, our support here at Harvard. I told you briefly about Corinne's work in the sort of ex vivo culturing and gene expression studies. Some of that was published last year. And then Henry has jumped ship and is, I guess, the first member of the lab to infiltrate Big Pharma. So I'm excited to see what he comes up with at Novartis. And then finally, I just want to point out that this is my farewell speech in Boston. So we're moving to UCSF in a couple weeks, and so we're looking for talented postdocs with experience in computational biology or more experimental approaches. So I would encourage you to check us out if you're interested in this area. So thanks.
Great talk, Peter. I enjoyed it. You know, as a microbiologist, when one finds an operon in one bug, one always wonders how widely it is spread. So we have organized the human microbiome in a number of these metagenomic species. So it might be interesting to see, you know, whether this operon or related homologs could be more broadly spread than just this one strain.
Yeah, I agree. I think that would be really interesting to look at especially your newer data with the larger sample set. I did some mining of the MetaHit data looking for Elenta, and didn't really see much of a signal of this gene. I think one of the issues with Elenta, which I didn't mention, is that it's found at not particularly high abundance in the gut microbiota. So we think the average cell count in the lumen of the gut is somewhere around 10 to the 7th cells per gram. So it's, in a total population size of 10 to the 12th or 10 to the 13th, it gets drowned out a lot in metagenomic analysis. But one of the things we've been trying to do is we've got some methods for doing enrichment culturing for Elenta so we can boost the abundance of the cells. And then we're also, using culture independent methods to try to do that.